I'm joined now by Dr. Pere Puigserver, who is getting the Outstanding Scientific Achievement Award. Congratulations on that.
Thank you very much.
I know you're giving this speech, Transcriptional Machineries that Control Glucose Homeostasis, the targets for diabetes. Can you give us a bit of an overview?
Absolutely. So what we have identified over the last 13 years is basically there is a connection between the nutrients and the hormonal signal, which is very important in diabetes, which is carried out basically through the two chief hormones, insulin and glucagon. So we have identified a set of transcriptional proteins which are in the nucleus of liver cells that are able to sense these nutrients, and as a consequence they can regulate expression of genes which are involved in glucose production. This glucose production is very important in diabetes because it exacerbates hyperglycemia that can lead to many pathological consequences. Furthermore, what we have used is a chemical approach using this information from these transcriptional complexes and have identified small molecules that can target these transcriptional machineries, allowing the control of the blood glucose, and as a consequence are important for diabetes.
Can you talk a little more in detail about the research on these metabolic circuitries and how they may translate into new therapies?
Yeah, so these metabolic circuitries are basically a set, as I said, a set of proteins which are sitting in the nucleus of the liver and basically are sensors for these nutrients and hormonal signalings. So these proteins are potentially be targeted with small molecules. Some of them we have identified or other ones. And as a consequence of this influence on the activities of these proteins, which by the way are enzymes called the acetylases and acetyltransferases. So as a consequence of being enzymes, they can be druggable, and manipulating the activity specifically in the liver, they can control blood glucose and as a consequence be potential therapies for diabetes.
That is certainly very exciting. So for attendees who might be hearing your talk, what do you hope that they take away from that and bring back potentially into their own clinical practice?
Yes, so I think it's difficult sometimes to be like a chief thing because in these attendees the audience is very broad. So you come from basic scientists to the clinical scientists. So, but what I would say is that we have identified a set of proteins that are important in the control of blood glucose production, and these proteins operate or function in normal conditions or the fat and fasting, but these proteins are dysregulated in diabetes. So as a consequence of being dysregulated in diabetes, it is possible to use chemical approaches as we have used to discover novel molecules and new drugs that can target this protein, and as a consequence can be potential new therapies for diabetic patients.
And that's what's so critical about this kind of research and these findings is that you can create new therapeutic targets. Thank you so much, Doctor.
Well, thank you very much.